I'm going to introduce our speaker today. Dr. Chunlei Su is an associate professor in the Department of Microbiology. Dr. Su holds a DVM from China Agricultural University, an MS in Animal Science from Brigham Young University, and a PhD in Pathobiology from Pennsylvania State University. He has been with the University of Tennessee since 2004, and his laboratory has been focusing on a large-scale molecular epidemiology and population genetic study of T. gondii. So with that, I give you Dr. Su.
All right, thank you, Dr. Miller. Let me share my screen. Can you see it?
Yes.
Okay, so thanks to UT One Health Initiative to give me the opportunity to present my work. So today I'm going to talk about the One Health approach to toxoplasmosis, and then from there rolling it into the next topic, which is developing a pathogen surveillance system. So I want to kind of share with what we did in Toxoplasma can be used to for the pathogen surveillance system. Okay, next slide. Okay, so since I'm a slow speaker, so I want to put the acknowledgement here first in case I don't have enough time to finish up. So Alyssa, probably you need to remind me when it's close to the time for me to stop. Okay, so first I would like to give an acknowledgement to a few group of researchers in the past 2 decades. We worked together on Toxoplasma epidemiology and population genetic studies, so we were able to get information to what we have today. So firstly, the USDA lab, Dr. Dubey and the associate there, and we have been working together for, say, almost 20 years. And other groups from different countries, like from University of São Paulo in Brazil, and University of Limoges in France, and the other lab from, say, WashU in St. Louis here in the state, and another group from China, and also Dr. Gerhold from our own campus here. So without those collaboration and many other collaborations, it's impossible to do this type of work. And also thanks to the funding agency, the American Heart Association, NIH, and also UTK seeds grants here, and to make it possible. Okay, so first, a little bit prime for Toxoplasma. Toxoplasma is a single cell parasite, eukaryotic parasite, which has to invade eukaryotic cells like human or animal cells to replicate and spread. So it is in the, say, phylum of Epicomplexa, and there are many protozoan parasites in this phylum, like Armeria and Sarcocystis, Plasmodium, the parasite causes malaria. And what is unique about Toxoplasma is it is a generalist. It can infect basically all kinds of animals, birds, mammals, all can be infected. And in its life stages, there are 3 stages. The fast replicating stage is called the tachyzoids. That's when there's no immune control. The parasite replicate inside cells almost freely and fast. So it's the tachyzoids. Here is to show the parasite inside the cell in cell culture. And then in host, when there's an immune response kick in, the parasite will be controlled, and those stay in the slow replicating state called bradyzoids. They can survive and then be transmitted. And the other stage is called the sexual stage, basically in feline, in cats, intestinal tract, in those epithelial cells. the parasite undergoes sexual replication by developing into gametes, and then the zygotes are shared in the feces and contaminate the environment, and then transmission can go on. So when we teach, say, the life cycle of Toxoplasma here, very often say we mention the two cycles. One is called the sylvatic life cycle, which means in wild animals, and the other one is domestic cycles, that means in domestic animals. But in general, you'll see, regardless, there are a group of definitive hosts, felines, the wild or domestic. They serve as the host for the sexual replication. So basically, that's in those cats, the oocyst being, say, produced, and the oocyst then can, say, being shared in the environment, contaminate the environment, and other animals can pick them up and get infected. So those other animals are called intermediate hosts because the parasite replicates asexually by dividing and multiplication. So then most of those intermediate hosts, they are resistant to the parasite, so they can, say, mount the immune response and control the parasite. So basically, the parasite form the tissue cyst, which is the chronic stage of replication. And among those intermediate hosts, when one animal eat another animal, and then the parasite can be transmitted. So there's called, say, transmission among the intermediate hosts. And if those intermediate hosts consumed by or eaten by the predator, say the cats, then the life cycle complete, and you generate more, say, oocysts from there to contaminate and continue the life cycle. Okay. So in terms of prevalence, in human, globally, it's about 30%. So in that sense, 1 in every 3 people is positive. Okay, so here is in general, the seroprevalence in human population. And the general idea here is in tropical region, the dark red, brown color here, those areas, the infection rate is very high, normally be above 50%. And here, if you see South America here, Brazil, it has, let's say, greater than 60% of infection there. And so in other regions, it depends. So in the States here, we are here, and currently the infection rate in the population is less than 10%. But in some European countries, the infection rate in human is pretty high, in the say 40 or so percentage there. That is due to their dietary habit. So if you consume those undercooked meat, then there's a potential to get infected. So the quick catch up here is we can get infected by consuming the oocyst from the say contaminated by the feline fecal feces, or we can get say contracted or infected by, say, eating undercooked meat. And then there's another way get infection, which is the vertical transmission, say during pregnancy, when the mother got infected for the first time, and there's no immune say protection there, and the parasite can spread quickly and then get through the placenta and infect the developing fetus. So that's the kind of the way human may get infected. So just as I mentioned, worldwide, the prevalence in human population is about 30%. And toxo infection is one of the major foodborne disease. So there was a study showing Toxoplasma infection account for the second most common cause in foodborne illness in the States. And so fortunately, for most infections, there's no symptom. So chronic infection. So initial infection, you may have a flu-like symptom and may not be even think about a toxic infection. But after a primary infection for about 2 weeks, the symptom will go away, and then the parasite will be controlled and then stay in the chronic infection state. And it is believed when we got infected, we have them, we have it, so we keep them. So for the life of our time. So, but occasionally there are small percentage of people can develop ocular toxoplasmosis. Basically, the parasite get into the eye and cause inflammation. It may, say, affect the vision there. And for immune compromised patients, particularly the late stage of AIDS, the parasite can reactivate and cause severe inflammation, and that can be fatal. So if not diagnosed quickly, that can be a problem. But we do see severe acute disseminated toxoplasmosis in human, but mostly reported in South America. And this may due to some atypical strains circulating in that environment, particularly the so-called Amazonia toxoplasmosis. So there are many reports in the Amazon region when people got infected, even though immune competent, there are many cases of severe symptom and organ failure and death of that consequence. So, and lately more say study on Toxoplasma from those areas recognize very often you see the so-called atypical strains. They are highly virulent to mice. So the virulence to mice may be related to the, say, this virulence in human infection. So how about in animals? It depends. So for most animals, they are resistant to the parasite. So they basically have the chronic infection, no symptoms, just fine. So in those group of animals, mostly birds, they can get infected, but mostly they are resistant for some reason. And the cattle are highly resistant, and the deer, the white-tailed deer, they are resistant. Rats, they are resistant, and the cats. And so even so for the white-tailed deer, I want to put a kind of a comments here. In white-tailed deer, toxoplasma infection is very common. And so we had some studies in Ohio or area, and in some region, you can see the infection rate up to 70%. We did a study here in Tennessee. It's about, say, 30% or so in those animals. They don't have a symptom at all. So for other animals like the Australian marsupials, the kangaroo, wallabies, they are sensitive, and the New World monkeys are very sensitive, and house mice, they are sensitive. So those animals, when they're infected, they tend to have higher mortality rate. And predators, the felines, mostly they are, say, resistant. So, but they have higher seroprevalence in general because they eat other animals and can, through the food chain, they can accumulate the parasite quickly. So in those animals, so for example, the bobcats, based on several studies in the States, their infection rate is close to 100%. So most of them get infected, carry the toxoplasma there. And so, but for chronic infection, very often you see the parasite in the muscle and the brain tissue. So if you want to isolate the parasites, you go to the heart tissue or get brain tissue from those animals. Okay, so what we have been working on, say, the Toxoplasma, say, molecular epidemiology and genetic studies for almost 2 decades. And so I want to share with you what we learned from those studies. And so talking about molecular epidemiology, population genetic studies, first I want to mention the methodology or the tools we normally have to do epidemiological studies. So the tools, you can divide it into 2 categories. One is called the detection. So the other one is called identification. So detection, infection, there are mostly two ways. One is use a serology test. So to find or to detect the antibodies. If the animals or human say have the antibodies, that suggests there was exposure and possibly, likely carry the parasite right now. And the other way is called to detect the DNA, to detect the DNA from the parasite. So the first one is serology test to look at the host. And you can also look for the DNA of the parasite in the host tissue. And for this, detection, normally you focus on those DNA sequences that have multiple copies. One example in Toxos field is the 529 element, which has about 300 copies. And the ITS or 18S ribosomal RNA gene, which has about 100 copies. And there's the other one is called the B1 gene, which has about 35 copies of that sequence. So because you have repetitive sequence, you have the increased sensitivity for detection. So, but detection only tells you you have the infection or you don't, but cannot give you more information in general. So there's this, another way, another tool, which is called identification or genotyping. So you want to genotype a particular strain and see what it is. So that gives you a higher resolution. And normally there are four kind of different ways, from multi-locus PCR-FLP method to multi-locus sequencing typing, MLST, multi-locus microsatellite genotyping, or the whole genome sequencing genotyping. So all based on what kind of questions you are asking and how much money you want to spend. So if you have lots of money, you can do the whole genome sequencing. But if you have, say, less, you can use the other method. So among those methods, basically they are based on two different mode of, mutation. So for the multi-locus PCR-FLP method, it's based on SNPs, single nucleotide polymorphism. So you see there are 3 sequences here. You see the mutations or different alleles here. And for RFLP, you find the restriction enzymes which can recognize those differences. So for this method, you can only recognize some of those differences, not all of them. If you keep them all, look at them at the sequence level, now you have the MLST. So you look at multiple loci and then look at the variation, that's MLST. And the other one is called microsatellite. Microsatellite, they are short repeats. So here in this example, that's 80 repeats. And those repeats, they generally have higher mutation rates, so give you better resolution in terms of identify different strains. Of course, the whole genome sequencing will give you the best resolution of all, but it is still expensive. All right, so from my lab, when we started this type of studies, we started with to develop the tool first. That was called multiplex nested PCR-FLP genotyping. Basically, you use two, nested PCR method. You have a first PCR to amplify all the markers you are going to do, and then you use that product serve as your template to do the individual markers, which give us a very good sensitivity. So the sensitivity for one reaction is about a 10 genome, equivalent. So that is pretty good in terms of sensitivity. So the reason we use this method is it is relatively cheap and almost every lab can do it. So when we started this study, we have it in mind. This type of study will need collaboration from from people all over the world. You don't want to make it too complicated and too difficult to do. So so everyone can participate and do the work. Okay. So here in this methodology, we have 10 genetic markers to work with, and you'll see they are, so for different strains, they have different patterns, and which can be used for a large number of samples at the same time. So we have a large gel format. We can run up to 150 samples per run. So if you have samples collected, it can be done quickly, relatively quickly, and give you the result. And we also adopted the microsatellite genotyping because it gives you the high resolution. So better than the RFLP there. So here is to show 15, say, microsatellite markers for each sample. So you see you see the variation over there. So we have 8 samples there. Each sample has 15 microsatellite markers. And so you cannot do much when you run the gel to look at it. And you have to put them in sequencing facility to get all the bands, say, revealed, and look at the size to determine the genotype. Okay, so with those tools in hand, we start to collaborate with people all over the world for over a decade. So doing the same thing, basically, and repeatedly use the same method. So eventually we were able to collect enough data to look at the genetic diversity across the globe. So in this study, we have almost, say, 1,000 samples checked. We identified 138 genotypes based on the 10 PCR-FLP markers and a few multi-locus sequence typing markers. So from here, we can roughly divide those samples into, say, 6 different clades. And here you see the diversity. So the same species, different strains with many different genotypes there. And the diversity, so then the question was, what is this diversity come from? And so we did the genetic, say, analysis showing that those diversity are largely generated by genetic cross. So you have a few, say, parental genome, and then they are shuffled among them frequently and to generate the diversity. Okay, so now if you put this diversity against the globe map, what do you see here? So this is another, say, study a few years later after we get the previous one, and we were able to look into 1,457 samples, and using the PCR-FLP method, we identified 189 genotypes globally. So if you look at the black dots on the map, those are the studies we did with collaborators, or the collaborators did with us to look at the results. So if you put them all together, look at the diversity genotype against the map, here is the impression. So first, if you look at the North Hemisphere, so the North Hemisphere, if you look at those samples, they have relatively low diversity. So the size of the, say, circle here indicating the sample size, and the pie, the pie here indicate different genotypes. So if you look at, say, from China, there are 62 samples, mostly belong to one genotype. They call it Chinese 1. And if you move to the, say, the West, you'll see in, say, the North Africa, Egypt here, over 100 samples, mostly type 2. And in Europe, mostly type 2. And North America, mostly type 2. And in South America, you do see type 2 over there, but it's less. But if you look at, say, Brazil here, it is diverse. There's no major dominant ones. So this is striking when we see the structure, the population structure here. So then the first question we want to ask here is, if you look at the type 2 strains, the dominant globe, well, they are widespread and dominant in many regions. And they also, well, so then the question is, can we kind of zoom in a little bit to see, can we identify, or at least, say, infer the origin of the most recent ancestor of those type 2 strains? So that's what we tried. So we look into the sample, we collected from those studies, and we got 322 type 2 strains. And so based on, well, the type 2 is based on the PCR-FLP method, and we now want to look at those samples with, say, higher, resolution markers, which are the 15 microsatellite markers. And we look at those samples by their, say, geographical distribution. So Europe, Africa, North America, South America, and see what they look like. So here is the population analysis. So the first analysis is within population diversity. So let's say we have 4 subpopulation, Africa, Europe, South Central America, and North America. And we have different number of samples. And then let's look at their microsatellite genotypes. So from here, we analyze the diversity of each group, subgroup. And from here, you'll see Africa, Europe, they have a relatively high diversity there. So that means this population, they tend to be older. So they have been there for a longer time, so they can accumulate those variation. And if you look at the North America samples here, their diversity is less. So that means they are more recent. So that's the within population comparison. Then we do the between population diversity comparison. So if you look at the, say, Africa sample with Europe, very close. There are very little variation. So Africa, say Europe, they are very close. But Africa and, say, Central and North America, they are quite different. So they are distant. And if you look at Europe and the South and North America, you see here the variation is less. That means the European isolates, they are closer to the South or North America samples. So basically, that's what you get from this genetic diversity analysis. So how about the genetic rooted phylogenetic tree? So not to trying to read anything here, just look at the color. The red means the Europe samples, and the blue is the, say, North America samples. So the rooted tree, the root is at the top, and if you think, imagine this is a tree, you put it down and look at the sideway and how they distribute it. So if you look at the red samples, they are basically from the root all the way down to the tip. So they are everywhere, and they are diverse, which makes sense from the population study, well, the within population studies there. And if you look at the North America samples, they are to the tip, and they are close to each other, which, yeah, we see the from the within population analysis. So what that inferred to us is likely the North America type 2 strains come from Europe some time ago. So that's the inference from the data analysis. Okay, so then let's get back to the big picture here. We saw, say, the North Hemisphere has low, diversity. South America has high diversity. But if you look at the data clearly, closely, you'll see, say, the Eurasian area seems like their variation is even less comparing to other areas. So the North America is in the middle. It does have variation, lots of variation, but they have the dominance of a few genotypes. And the contrast here in South America is totally diverse. So then we thought, well, what happens? Potentially we can see through the data from the North America samples, because it has the intermediate level of, say, diversity there. Can we see how the genotypes distribute in those areas? So that's what we did. We dig out, say, 623 samples from North America, and then look at their genotype, say, distribution by space. So we divided those samples into 3 categories. One is called farm-bound. So the animals are on farm or close to farm, and we collected those samples. And we see somewhere in between. So the goat and the sheep, which are often on the pasture or between the forest and the farm. And we also look at the samples from the wild and then look at the distribution of genotypes. So when we look at this genotype distribution, we see on-farm or farm-bound, they are mostly dominated by the type 2 strains. Remember, the type 2 we thought probably come from Europe. And so on farm, you normally see the dominance of type 2. When you are in between, you see type 2 decreased, diversity increased. And when you go to the forest, you see lots of diversity, type 2 decreased even more. But interesting, there's another type called type 12 dominant in the wild animals. That's a very unique phenomenon in North America. So what we can say, infer from this study, it seems like the type 2, let's say the domesticated one, are penetrating through the space into the wild animals. So we are spreading the, let's say, domesticated type 2 into the wild environment there. Okay. So if we look at those distribution in the terms of virulent strain, remember the North Hemisphere has less variation, and the dominant, say, genotypes in North Hemisphere, they are non-lethal to house mice. So they are intermediate to non-virulent in a sense. But in South America, you see a lot of highly virulent Toxoplasma genotypes and strains. So why it is that way? So we are trying to understand that by looking back into the life cycle. And we remember we're talking about, say, the life cycle involved the sylvatic cycle in wild animals and domestic samples in, say, cats and other animals. And so does the virulence, say, affect the transmission of those parasites? Remember, in the domestic cycle, we are closely associated with the house mice. So if you go to the farms, every farms you will find house mice because they are domesticated, well, kind of domesticated, unwillingly in history and to become associated with human. And now you have cats and mice. And if the parasite is highly virulent to the mice, it may not be good. But in the wild environment, the wild animals, mostly they are resistant to Toxoplasma, so they can handle those virulent strains. Maybe that's something beneficial. So we have the, then we dig into the literature, and to see that in literature, we see that in experiments, there are many published data showing that if you put all those so-called non-lethal strain into naive mice, in general, they establish chronic infection and the transmission is not a problem. But for those highly virulent lethal strain, if you infect naive mice, normally they kill them within about 2 to 3 weeks. However, if you inject those strain into chronically infected with non-lethal strain into those mice, the lethal strain can actually super infect. So now the mice can have 2 different strains simultaneously. And then for that case, the lethal strain can be transmitted from those chronically infected mice. So there are 2 factors, virulence and super infection capability. So can we say trying to see if we play with those 2 factors, can we see anything there? So we cannot do without doing the lab work will be more difficult. Let's try to simulate the 2 scenario. One is called a domestic cycle. You have highly virulent strain, you have intermediate virulent strain, you have non-virulent strain. Put them into the, say, mice and cats in that environment and let them circulate, simulate for a while, 20 years, 50 years, or 100 years, see what will happen, And here from the domestic cycle, we see the highly virulent strain, they basically lost in the environment, in the circulation. But in the sylvatic cycle, because the animals are highly resistant to even highly, say, a virulent strain, so they can survive. But because they have the capacity of super infection, maybe they have the advantage of that. So in this case, you do see the highly virulent strain will be there. And okay, so how to put all things together? So we start to think about, okay, we have the domestic cycle. The domestic cycle only, say, occur or started in relatively recent time. If you think about before agriculture, we don't have the, say, domestic cycle. So in that time, so let's get back 10,000 years ago, it's basically white cats and all the other animals, wild animals. So this was the cycle of Toxoplasma infection. However, agriculture started about 10,000 years ago. So we know the Fertile Crescent in the Middle East area, that's the place the oldest agriculture started. And about 11,000 years ago, when that started, the small rodent population increased dramatically based on the studies, published studies. And with that, the cats at that time was wild cats. They like to see all those rodents. So they start to get close to human and gradually become the pets we have. And shortly after that, the initial, say, rise of agriculture, the second one, oldest one is in China, in the, say, Asian area, where the rice start to be, say, domesticated, and also cats start to kind of increase there. And it was only recently, last 500 years, when, say, Europeans migrate to the Americas, brought the wheat, that was the major, say, crops from there, and also the cats, and unwillingly those mice. So if you look at the, say, radiation of house mice, they start from, say, the Eurasia area and now get into North America, and you see them everywhere in North America and also in the peripheral area of South America. Okay, so that gradually give us the domestic cycle. And so in the domestic cycle, there's a pressure to select against the highly virulent strain, but in the wild, you still have those highly virulent strain there, particularly so in South America. Okay, so all this may kind of overlap, while understandably make sense that when the human population increase and agriculture spreading, and the domestic cats and the mice, and that probably reduced the diversity of the Toxoplasma and also reduced their virulence. So here the diversity reduced, and comparing to South America, and also probably the virulence being reduced in those scenarios. Okay, so that's about the diversity and how the epidemiology and the diversity and how we maybe learn from that process. And so I want to say, emphasize a little bit here about the Toxoplasma. The oocyst is the main source of contamination. And in the study, in the past study in Australia on the island, in the beginning, there are lots of, say, infection in the sheep on one island, but when they removed the cats, the oocyst disappear, and then the infection in sheep also dropped significantly. So the oocyst is the big player in terms of the transmission of the parasite, and we have cats, all kinds of cats in the wild. They share the oocyst, contaminate the environment, so the intermediate host got infected, and the livestock can get infected, and those parasite oocysts can be washed to the, say, the sea and all the water to contaminate the, say, the marine animals, and also can contaminate the water and food to human. And a few years ago, 2018, there was a huge outbreak of Toxoplasmosis in Santa Maria, Brazil. In that, infection outbreak, more than 900 people confirmed infected, including 135 pregnancies, 3 fetal deaths, several, say 9 abortions, and 28 congenital toxoplasmosis. This is so far the biggest outbreak in the world. And later studies suggesting the contaminated water be the potential cause of infection. So in the environment, when we look at the seroprevalence often is a good indicator of infection rates. And in domestic cats, there's a study indicating they're about 37%. That's pretty high. And in wild cats, it's even higher. So yeah, well, remember the bobcats, their infection rate is close to 90, 100%. So lots of, say, infection over there. So what we can do about this? So we do need to have a surveillance, say, a system for this case, for Toxoplasma, and particularly for livestock. And we all know the organic farming is getting more and more, say, popular and more appreciated. And in that case, the animals tend to, say, being exposed to more Toxoplasma in this process. So we do need to, say, do surveillance on that, and the seroprevalence serology test will be a good one to start. And in our lab, we have the capacity to produce antigens to do this, to support this test. We also are able to isolate the parasite and do further studies and to track outbreaks. And also we are in the process to establish a repository of Toxoplasma strains and make it available for research, labs. So, well, I'm running pretty low on time. So a quick kind of a mention of a recent study from Toxoplasma outbreaks in the zoo in Florida. And from 2014 to 2018, they have multiple deaths of the, say, kangaroos and wallabies. And we were able to collect those tissues and do either, say, isolating of the parasite or genotype the parasite using the animal tissue directly. So from here, you can see in the 2014, year 2014, we identified one genotype called 263 from that, say, outbreak. And later, say, from 2015 to 2018, we do have a few samples spread out through the years, and we identified type number 2. And in the, say, end of 2017, we identified number 2016. What does that mean? That means there are, say, multiple outbreaks of Toxoplasmosis in the in those animals. It's not a single type of genotypes. But from here, you can type 2 spread pretty wild. That means the type 2 may be cause the outbreak in separate time. So maybe even though they are the same genotype, they may not be the same batch of, say, oocyst there. So provide more information on the parasite and may help us, well, help to manage the runoff from rains, so you can avoid those oocysts being accumulated to the site of those animals. Okay, so future studies, just to quickly mention that we know very little about chronic toxoplasmosis and other human diseases. Either these, well, in literature, you can find some, say, reports about the toxo infection with human, say, disease, mental health, heart disease, or co-infection of toxo with other diseases. There are reports showing up to see, okay, toxo infection may affect those. Okay, so I have a few minutes to talk about the second part of it, which is to develop a system for molecular detection and identification of zoonotic pathogens. And this study is supported by our UT One Health Initiative seed grant from last year, and with a group of investigators here on different campuses here, and with our student and technician And I say, so first, thanks to everyone, we can get this one started. Okay, so the goal here, what do we want to do? We want to detect and identify zoonotic pathogens in animals. So zoonotic pathogens, there are many different types. And so we are trying to go beyond, say, one pathogen. And so what do we need? We will need to detect novel and known pathogens all together in one place. And so this is necessary for long-term epidemiological studies and surveillance of zoonotic pathogens. So given the current time, we know we went through and we need to have a better surveillance system. So for this type of work, what do we want to know? So in this system, what do we need to have? And there are three significant components we need to have. The first one is we need to collect all those biological samples, and we need to establish our biobank. So those samples need to be collected, and people need to know we have it, and that's say a very important biobank. Without those samples, the samples should be collected with the breadth of the timing, so different times and different locations. You need this kind of two dimension. Both need to be there. So that's one aspect of it. And we did collect it as a group, a few 300 samples so far, and put it into a database, online database there. And also we need to isolate pathogens if possible. So for this, we have a good collection of toxo strains, and we are supported by NIH grant trying to make a repository from there. And this can also be applied to any other pathogens we are interested in. And the other objective tool here is to develop a molecular method so we can detect and identify the pathogens. And from there, we can use the two parallel approach. One is metagenome sequencing to identify known and unknown pathogens. And in the meantime, we do want to detect those major pathogens we want to look at. So for that, we are trying to use the targeted sequencing. So PCR-based targeted sequencing. And when you have both, put them in the, a genome sequencer, the next generation sequencing machine, and then see what we have. So what kind of pathogen you want to target at? We look into the literature and see the 2017 U.S. One Health Zoonotic Disease Priority Report. This report was generated by CDC, USDA, and the Department of Interior. So they were 56 pathogens, zoonotic pathogens listed here. And I kind of put a part of them here, the top, 27 here. And you see mostly they are viruses. So virus, bacterial, protozoan parasite, fungi, they are all there. So in that list. So if you want to look at those pathogens, what kind of method or approach you need to go with? So we look into the literature and see all available methodology there and how to integrate them together. So remember, there are different, category of pathogens. They have different biological characteristics, and you cannot just bring them, mix them together and do it. So what we need to do is we have three kind of parallel approach. One is for viruses. So for virus, we need to use a methodology to enrich, viral, nucleic acids from the host tissue, from host, nucleic acids. So there are methodology for that. And for bacteria and fungi, there are methodology to enrich their, DNA from the host genome. And we kind of adopt those methods. And then we take the whole took the everything in one pot approach. So for example, for those protozoan parasite, they are tissue residing parasite. It's very difficult to separate them from the host tissue. So what we do is we extract the total DNA from all of them, and then from there you do the targeted, say, sequencing. Use targeted PCR and sequencing them. So now we have the three parallel, approach there, and at the end, well, you have to put adapters to those, say, sequences, and eventually feed them into the whole genome sequencing, say, machine, and then generate the data, and from there look at the what you have. So let me see. I have a couple of minutes to quickly finish up. So, well, here is a primer design. No need to look into the detail. That we use all the different adapters. So eventually you can put those sequence into the next gen sequencing machine. So we put a number of positive controls there, including positive, negative, gram-negative bacteria, gram-positive bacteria, fungi, parasite, viruses there. So those are the, say, benchmark we need to look, and then we included a few samples from our collection and see how this methodology work. So here is the quality control of those, say, library preparation, and they seems the samples are all good, within the right range in terms of amplified sequences. And for the positive control, we have all the sequences amplified. And for negative control, we have two. You can see one is contaminated with something significant here, which we figured out was Toxoplasma. So, well, we have lots of Toxoplasma contamination there. But for the other negative control, you'll see the smear there. That means there are some background DNA or say nucleic acids there, which serve as a baseline to say to evaluate the reagents, the procedure, everything. Okay, so I have to be quick. So here, when you have the data, what are you going to do? And we were kind of scratch our heads how we analyze those data. And by searching, say, online, we are fortunate to see the Chan Zuckerberg Foundation. They are putting a team of people, bioinformatic people, to generate a pipeline. You basically can drop your whole genome sequence data, and they will give you the results. So that's what we did, and look at all the positive samples. We had 13 positive samples, and we can detect 12 of them. So those stars indicating those, say, strains we put in, and they showed up. And here to say E. coli, we have a very good, say, genome coverage over there, and the Listeria, very good coverage. And there's a spike, that's the 16S ribosomal RNA, say, targeted sequencing. And the Cryptococcus, you see, have some coverage of the genome. And Toxoplasma, we have it's a big genome, so you have less reads, but we do have a decent coverage there. And also to see the 18S ribosomal RNA being targeted sequencing and showed up. And we do have the human herpes virus, which is a DNA virus, showed up. And from some of our samples we used to isolate Toxoplasma in the past, we did see, we had positive isolation, and we did see the signal of Toxoplasma. We also see other, say, species, Sarcocystids species identified. Those are the tissue residing parasite. And from one bat sample, we also see a parasite, Nephrio iosospora, which is, say, a parasite known to infect the kidney of animals, and we saw it there. So we did not see our coronavirus, bovine coronavirus, as a say, a spike in, but when we look at the when we use the nested PCR, we do see those signals showed up there. So there's some more work to optimize those samples. Okay, I think I'm running out of time, so I will stop here and for questions. Thank you very much.
Thank you. That was awesome. That was a wonderful presentation. Do we have any questions this time?
Nothing in the chat. Feel free to use the chat or unmute yourself if you have a question for Dr. Su.
And I do have one. Maybe I missed it and you touched on it. Survivability of the organism in salt water versus freshwater, since we're seeing it in marine mammals. Is there, like, do you know what the difference is? Is it equal?
You mean in the in Toxoplasma or?
Yes, for Toxoplasma.
Yeah, Toxoplasma, the oocyst is very resistant to chemicals and salts. So in experiment, if you collect oocyst from cat feces, you can use 2% sulfuric acid, or you can use the 6% the bleach to treat the sample, and the oocysts are just fine, and everything else being gone. So, and in salt water, yeah, they can stay there for a long time. So very resistant. So the oocysts only sensitive to temperature. Yeah, freezing or heating can kill them quickly, but otherwise they are very resistant. Yeah.
So just follow up with that. Given that we are seeing this be impacting marine mammals, marine reptiles, et cetera, that are far out in the ocean, or marine birds rather, what do you think is, how do you think they're getting it? I mean, we kind of surmise that maybe with manatees they're getting it while they're eating in the seagrass. But what about like some of the dolphins, and what are some other ways that you think they might be getting it?
Yeah, we do see infection in dolphin. We actually get isolate from dolphins too. Seems like they are okay. Well, at least to some degree they are resistant, but I think seal might be sensitive. Yeah, seal might be sensitive. I remember see the report from Hawaii. They have animal died of Toxoplasma infection over there. Yeah. Yeah. So technically, in experimental study, people are looking at the vaccine for cats. If you can vaccinate cats to stop them shedding oocysts, you may reduce the infection rate in the animal population. Yeah.
Oh, that would be awesome.
Yeah. Yeah.
Any other questions? All right. Thank you so much.